because my wife never smiled when she was that pregnant. It's just not, I don't know how much they paid her to smile like that, but you're not that happy when you're that far along. At least my wife wasn't. Okay, so sexual reproduction. Obviously, we're not going to spend most of the time talking about the birds and the bees. Hopefully, you already know everything regarding that. But what we are going to talk about is when any organism, not just humans, but when any organism that undergoes sexual reproduction creates the gametes, the sperm and the egg, what is it that is so special that makes sexual reproduction actually more advantageous for evolutionary survival than asexual reproduction, which is the cloning process? We're going to talk about why it is that sexually reproducing species do a better job of surviving as a whole, because ultimately sexual reproduction is not a cloning process. It's a way of taking two sets of genetics and scrambling them up so that the offspring is always unique. Okay? So that's why sex. Okay? Not beyond the normal reasons But that's why organisms that are predominant in this world are sexually reproducing, because of the amount of genetic variation that is propagated from one generation to the next. So here's why. This is why sexually reproducing species have a greater advantage. When you have a clone, it takes a long time to generate any type of genetic diversity, because the only way in which you can get typically any type of new diversity is through random mutations. And I mean random, meaning it's not directed. They happen due to the environment, to various things. You're not going to get a lot of diversity. Now, this is good for us because when we get an infection, say of a bacteria, this ecological disaster can be just the taking of an antibiotic. We're assuming that because all of these bacteria are all clones of one another, that a single shot of this one type of antibiotic is going to kill them all off, and we're usually right. We're usually able to kill most all of them off. But when you have an organism that the population has a great amount of diversity, then one single change in the environment is not enough to kill the whole species off. And the reason for that is because usually there is enough variation within the species to adapt and survive. And this is why sexually reproducing species have a greater advantage. So we'll talk more about this when we get to evolution, when we get to ecology, but this is, we see this all the time, that when environmental factors change, food sources change, temperature or other types of parameters change, if the species can't adapt, then they die off. Okay? And 99.9% of the species that have lived on this planet are dead, according to our fossil records. So ultimately, it's common for many species to go the way of the dodo, so to speak. However, this is advantageous primarily because even what we might consider problematic genes, like cystic fibrosis, sickle cell anemia, Tay-Sachs disease, these diseases we've been talking about, even though they're problematic, in the right conditions, they can actually save your life. We'll show how individuals who have or carry some of these diseases are protected against things like malaria and typhoid fever and cholera and even tuberculosis and things of that sort. So there's a reason why some of these things persist in the human population, even though we're like, well, why are these diseases not eradicated? Well, sometimes nature says, hey, this will do you good. It does a body good, right? So that's why sex. Because ultimately, any species that reproduces sexually produces mass amounts of genetic diversity. And that genetic diversity is what gives them an evolutionary advantage for survival and reproductive success. So we call this process of gamete formation meiosis. And you're going to see that it's very similar to mitosis. It's got some of the same phases: prophase, metaphase, anaphase, telophase. Okay? But what happens during those phases is very different, especially in the first set of processes in meiosis. Now, in sexually reproducing species, unlike mitosis, when you produce the gametes, you have to split the genetic information in half. Why do you think that's the case? Why can't you just, let's say you have two parents with two chromosomes, why can't you just fuse those together? What will happen? Yeah, it goes from there, and then it'll go, 184 and so on and so forth. You can't. In order to maintain the constant nature, which is very tightly controlled by our cells, the same number of chromosomes, the same number of genetic information, we have to split our 46 chromosomes in half and ultimately pass on one of all of the sets of chromosomes that we have. Now, we don't split them in half in that we cut them in half. Remember, this is a karyotype of the human genome. We have 2 of every type of gene. Well, during meiosis, what happens is we only pass on 1 of those 2. And it's random which one we pass on. Now remember that these are the chromosomes you inherited from your parents. So if you inherited this one from your mother, you inherited that one from your father. If you inherited this one from your father, you inherited that one from your mother. And when you pass your genes on, you might pass on your mother's chromosome number 17, but your father's chromosome number 18. And this is how your offspring becomes a combination of your parents and their parents and so on and so forth. This is where the genetic mixing and matching comes into play. Now, most cells in your body do not undergo meiosis. Only a few select cells actually do this. So like we mentioned, those somatic cells, which are the body cells, these undergo mitosis. If they divide, which your adult stem cells do to regenerate your tissues, it's mitosis. But in the sex organs, the testes and the ovaries, this is where you have what are called the germline cells. These are special stem cells that have the ability to undergo meiosis. So what ends up happening is the parent cell, remember we call it diploid because it's got 2 sets of all of our chromosomes. So technically speaking, you can look at it like this. 23 times 2. We have 2 sets of all of these chromosomes. When it goes through the process of meiosis, it essentially only takes 1 of each of those sets, and when you create the gametes, the cells are now haploid at the end. They only have 1 of each of the chromosomes, and that includes the sex chromosomes. Each sperm and egg is essentially going to have 1 sex chromosome to it. And then when the sperm and the egg come together, you reconstitute those 2 sex chromosomes. Now for women, they have 2 X chromosomes, so they're going to pass on 1 of their X chromosomes. For men, that's where it becomes a 50-50 shot. Half of the sperm have the Y chromosome, half of the sperm have the X chromosome, and so if the sperm with the Y chromosome fertilizes the egg, you're going to have a boy. If the sperm with the X chromosome fertilizes the egg, you're going to have a girl. So it really is the guy's fault on the sex of the child. I shouldn't say fault. It's the guy's sperm that ultimately determines what the sex of the child is going to be. So, the life cycle of us, primarily from the moment of fertilization, when that zygote forms, and you form that diploid cell, it's all mitosis. It's all mitosis from the formation of the fetus to the baby to the adult. But when you reach certain stages, that's when meiosis occurs. Now I'm going to test you on this next concept. A lot of time people don't understand the difference in sex cycles in males and females. Now we're primarily going to focus on humans. Because when you look at other organisms like plants and frogs, they can be similar to us, like frogs, or they can be really different, like plants. So even though plants undergo sexual reproduction, the stages that they go through, they're just kind of all over the place. So we're just going to focus on human sex cycles and things of that sort. I mean, you can go further into it, and I think your book covers a number of different things, but I choose just to primarily focus on humans. Now, here are some facts that I want to test you on, because a lot of people don't know this. When women are in the development stage as a fetus, before they're born, the germline cells, these stem cells that produce the eggs, will actually begin meiosis before they're even born. Okay, so before they leave the mother's uterus and whatnot, they are creating all of the oocytes, which are the eggs, and they are beginning meiosis before they're even born. So women will create all of the eggs that they'll ever have for their lifetime before they're born, and then those eggs will begin meiosis before they're born. Now, let's say they start off with like 2 million. It might be as many as 4 million, but let's say 2 million or whatnot. By the time that women reach sexual maturity and they start ovulating the eggs, they'll have lost about 3/4 of them, and they'll only have about 500,000 left or so. Okay, so most of those are just going to die off essentially during the infant stages before they reach sexual maturity. Now, once they reach sexual maturity, cells will keep dying, but some will mature and essentially go through the ovulation process. Once a woman reaches her 40s or whatnot, mid to late 40s, when she runs out, that's when menopause hits. And so there's only a certain window in which women are fertile to be able to have a baby. Now, men, on the other hand, they don't even start producing sperm until they reach sexual maturity. They have the cells in place, they're ready to go in the testes, but they will not start producing sperm until they reach sexual maturity, and then they'll just produce sperm for the rest of their life, essentially, until they die. So, I mean, there may come a point where it shuts off because of hormonal levels or whatnot, but pretty much they produce sperm throughout the rest of their life. So, that's the fundamentals of the different reproductive cycles. Women have this window in which they can ultimately have offspring. Men have a much bigger window, but that's the difference is when meiosis essentially occurs. Here's another fascinating thing, and I'm going to show it later, but when a woman ovulates an oocyte and it starts traveling down to be fertilized, it still hasn't finished meiosis. It's still kind of right in the middle of the second stages of meiosis, and it won't finish it until fertilization occurs. If fertilization doesn't occur, then it will just go straight through, and that's when you get the menses and whatnot. But ultimately, meiosis is almost, a huge lifelong process in women. In men, they're just pumping out millions of sperm every day. So, big differences between when meiosis occurs in males and females. Now, let's talk a little bit about some of the mechanics of meiosis and why the cells are so different when they go through this process of meiosis, where they're not genetically identical like you would get in cells undergoing mitosis. Now, here's the first difference. Meiosis has 2 rounds of cell division. Not 1, but 2. So we usually distinguish them by saying meiosis 1 and meiosis 2. Meiosis 1 is obviously the first round. They got it right this time, instead of, photosystem 2 and 1 and all that kind of stuff. So meiosis 1 is the first round. Meiosis 2 is the second round. We're not going to talk too much about meiosis 2. The reason for that is because this process is almost identical to mitosis, and it's merely just kind of a finishing off process. All of the genetic recombination that we're going to focus on happens during the first round of meiosis. So that's where we're going to spend most of our time, and that's where we're going to have most of our questions. Okay? Meiosis 2, there is no genetic recombination going on during that process. So that's why we're going to focus on meiosis 1. That's where all the shuffling occurs, all of the mixing and matching of what your parents gave you to pass on to the next generation. Now, when you look at the chromosomes, we have 2 divisions of chromosomes. We have what are called autosomes. These are the non-sex chromosomes. Okay? We have 23 pairs of chromosomes, so we have 22 pairs of autosomes, and then 1 pair of sex chromosomes. So you're going to learn in the next lecture why this is important, because we have autosomal disorders and then we have sex-linked disorders. There's a difference between where the genes are found on which chromosomes and what effect that they have depending upon your sex, or whether it doesn't matter what your sex is in terms of the autosomes. So it doesn't matter whether you're male or female, all males and females, all humans, have the same groupings of genes on the same chromosomes from 1 to 22. For example, if the insulin gene is on chromosome 17, it's found there in both males and females. If you have hemoglobin here on 13, it's found in males and females, and so on and so forth. The genes are pretty much in the same place, no matter where you look. So the main difference between males and females comes down to whether you have two X chromosomes or an X and a Y. Now, this is not the same for all species. In some species, it's the females that have different chromosomes, and it's the males that have the same chromosomes. So don't think that this applies to all life in that females always have the same types of chromosomes and males have different types. It's different for different organisms. Okay, so notice the Y chromosome is so much smaller. There's only about 100 genes on this little chromosome, but that makes all the difference in terms of sexual maturity and identity and things of that sort. The X chromosome, there are over 1,000 genes here. Now, not all of these have anything to do with sex development. So just because you say X chromosome, for example, there are genes here that encode the cones, the proteins for your retina in your eye. They have nothing to do whether you're male or female. So there's even a blood clotting one on this one as well. So not all the genes on the X chromosome have to do with sex development, okay? But all of the genes on the Y chromosome do. So in this case, the Y chromosome makes all the difference between that. Now, just in case you think it's unfair, we'll talk a little bit later on how women, they do have two X chromosomes, but at a certain point in development, one becomes inactivated. So women, in all of your cells, you only have one active X chromosome. Men, you have the X chromosome and the Y chromosome active. So even women essentially have only one of their X chromosomes is really going to be active in their cells for their entire life. You don't need two. You only need two in the initial stages of development, and then one of them just shuts down randomly. Okay, now let's talk about the difference between sister chromatids and homologous chromosomes. This is a part that if you don't get this concept, you're going to have a difficult time a little bit later on. Now, we've talked about sister chromatids before. When we were talking about mitosis, we talked about how when chromosomes duplicate during semi-conservative DNA replication, each of the chromosomes ends up being copied. Well, that's the same thing for these chromosomes. It's no different in mitosis or meiosis. So whether the cell is getting ready for mitosis or whether the cell is getting ready for meiosis, doesn't matter. All of the genetic information gets copied. Okay, so you could say, for mitosis, you have 46 chromosomes, each one of them gets copied, and when the cell splits through mitosis, then you essentially split the sister chromatids. Each cell gets one of the copies. Now, in meiosis, the same thing starts off. We have 46 chromosomes, and all of those chromosomes get copied. And so each one has sister chromatids. Now, they've done a good job of color coding these for pink for your mother and blue for your father. Whoever came up with those colors, I don't know. But those stuck or whatnot. Ultimately, because we have two of every chromosome, one from each parent, we call them homologous chromosomes. Okay, so there's a big difference between sister chromatids and homologous chromosomes. Sister chromatids are carbon copies of each other. For example, here's a chromosome from your mother, and when your cell gets ready for either mitosis or meiosis, doesn't matter, then it's going to copy that one, and then they're going to be held together by a centromere. So that your mother's chromosome for that chromosome pair gets copied. Well, here's your father's chromosome that he gave you. That one also gets copied. So each one respectively has their own sister chromatids. These two are identical to each other, and these two are identical to each other. But what about the pairs? Well, they're not necessarily identical to each other. You've received these chromosomes from separate parents, and those parents have had their parents and their parents and so on and so forth. And since mutations are always occurring, which are changes in the genetics, even though your pairs of chromosomes have the same types of genes, for example, let's say that this B gene is for hair color, and let's say this is for brown hair, and let's say this is for blonde hair. they're both a gene that encodes the same thing. They're found in the same location on your homologous chromosomes. They both encode the same, or supposed to encode, the same type of gene. The protein for hair color, or maybe the protein for eye color. Maybe that's this one right here. Well, here's the difference. Maybe your father, or his great-grandfather had a mutation that changed the nucleic acid sequence of that gene. So ultimately what happens is, though homologous chromosomes have the same types of genes on them, the exact nucleic acid sequences might not be the same. What do we call those? We call them alleles. Okay? In fact, Gregor Mendel is the one that coined this phrase back in the 1800s, before they even knew anything about genes and things like that. But alleles. Alleles are the same type of gene, but different versions due to genetic mutations. Okay? That's why there's so much differences. That's why there's so many different hair colors and eye colors and skin colors and different heights and different intelligences, different blood types. You know, for example, blood type is one gene. We call it the ABO gene. But there are multiple versions of that gene. Some of us have the A version. Some of us have the B version. Some of us have one of each. And you might be blood type AB. Or you might have the O version. And that's where blood type comes into play. So one gene can actually have multiple alleles to it. All right, so what are homologous chromosomes? They have the same size. So look at the karyotype here. These two are homologous. They're the same size. They have the same types of genes on them. They have the exact same amount of genetic information, which is pretty much saying the same thing as they're the same size. And you get one from each parent. If you got this one from your mother, you got that one from your father. But the alleles might not be exactly the same. Your mother may have passed on the brown hair allele to you, and your father may have passed on a mutated version, the blonde hair allele of that gene. And so on and so forth all the way down the line. Your mother may have passed on the O allele for blood type, but your father passed on the A allele for blood type. And so you're going to be the combination of those two alleles. Okay, so homologous chromosomes have the same size, shape, construction, but the alleles may be different. Now, some alleles might be the same. Maybe both parents pass on blue eyes. Okay? Maybe both parents pass on blood type A. That's possible. So the alleles can be the same, but they don't have to be. They're not carbon copies of each other. So that's the difference between sister chromatids and homologous chromosomes. All right? So what ends up happening, now I can explain meiosis in a nutshell. What ends up happening is in the first round of meiosis, homologous chromosomes separate from one another, while the sister chromatids remain attached. So what ends up happening is on each of these chromosome pairs, one of your mother's chromosome will go into one cell with its sister chromatid, so the pink one right over there will go into one cell, and the blue one will go into the other cell with its sister chromatid. In the second round of meiosis, that's when sister chromatids pull apart from one another. That's meiosis. And that's why it has to go through 2 rounds. So in the first round, all of these pairs split up. Now let's look at the sex chromosomes. For males, the X chromosome, which you inherited from your mother, guys, because you're a guy, because your father gave you his Y chromosome. The X chromosome with its copy, its sister chromatids, it goes into one cell while the Y chromosome goes into the other cell. And then during the second round, the sister chromatids of the Y chromosome split up, and the sister chromatids of the X chromosome split up. So it's very easy to visualize it when you're dealing with male sex chromosomes, because these are the only non-homologous pair of chromosomes. Why are these non-homologous? What did we say about homologous chromosomes? Same size, have the same types of genes. These do not have the same types of genes on them. The X and the Y chromosome have totally different genes from one another. Now, women do have homologous sex chromosomes. Because the X chromosomes are the same size, they do have the same types of genes all the way down the line. They've got the same types of genes in the exact same locations. The alleles might be different though. Just like all of the other chromosomes, you've got one X chromosome from your mother and the other X chromosome from your father, and the alleles might be different. Okay? So we'll finish off on this concept right here. They start out diploid. After the first round of meiosis, they become haploid. We haven't even gotten into the little nitty-gritties of this. We'll cover that next week. After the first round of meiosis, the homologous chromosomes are separated from one another to where each cell is now haploid. Then through meiosis 2, that's when the sister chromatids separate from one another, and they're still haploid. Remember, sister chromatids are exact copies of that original chromosome. So when you split them up, you still only have, those two chromosomes in this scenario. Now the phases are the same. You've got prophase, metaphase, anaphase, and telophase. And since there are 2 stages of meiosis, then we also give the phases a 1 or a 2, depending upon which phase they're in. So if it's part of meiosis 1, then we just say prophase 1. And you'll see this on some of your quiz questions as well, where it'll ask you what will be going on during each stage. If it says prophase 1, that's prophase of meiosis 1. If it says prophase 2, then it's prophase of meiosis 2. As I mentioned, most of what we're going to go over is in meiosis 1, but there will be some things that we'll talk about, especially with anaphase 2, that become important in one of the concepts we're going to discuss. So you will need to just know that I'm not going to say prophase of meiosis 1, we'll just say prophase 1. Okay. Now, like mitosis, you got some of the same things going on in the early stages of prophase as you do with meiosis. So what were those things that happen in prophase of mitosis that we already know, that we already learned? What were some of the things? What happens to the chromatin? It becomes what? Remember? Chromatin what? Condenses. Okay, so chromatin condenses. Again, you have to be able to manage and move the chromosomes around, so they have to supercoil down to 1,000th of their original length. What about the nucleus? What happens? What happens to the nuclear envelope? What's that? It dissolves. Essentially, it breaks apart. And the same thing happens here in this prophase. And what's the third thing that typically happens? What do you see here? What forms? The mitotic spindle. Okay. Same 3 things happen here in prophase. Okay? But that's not what we're concerned about with this chapter. What we mainly want to look at is what's different? What makes this unique? Well, there are actually 2 different things that occur in prophase 1 that are unique to meiosis that you will not find in mitosis. Well, here they are. The first one is, remember we talked about homologous chromosomes. Those are the pairs of chromosomes you get from your parents when you were formed from the fertilization of the sperm and the egg. So remember, homologous chromosomes are the same size, they have the same shape, same types of genes, but the alleles might be different because you inherited them from 2 separate parents. And so therefore, mutations down their line ultimately arrive at you. Sometimes you have the same alleles. Both your parents pass on the blood type O, and sometimes you have different alleles. One parent passes on the allele for blonde hair, the other passes on the allele for brown hair. So, homologous chromosomes pair up during prophase 1. That is a key aspect of prophase 1. So what that means is, let's look at the karyotype here. Each of these homologous pairs will find each other. Okay, so chromosome 17 will find each other. Chromosomes 18, 21, 22, including the sex chromosomes find one another. Now here's the only exception to this rule. The male sex chromosomes, though they are not homologous, still find one another. They still pair up. So that's the only exception to what goes on. But all of the pairs, the 23 pairs of chromosomes that you have, the homologous pairs, all of them find their respective pair. That's the first thing. So the question becomes why? Why do they pair up with one another? Because that's crucial to this. This is what we call the first recombination event of 3 that you're going to be tested on, that you're going to have to know. So the first one is called crossing over. Now, has nothing to do with dying and going to the other side or anything like that, but it has everything to do with genetic recombination of alleles. So here's what crossing over happens. So let's look at this scenario. Here's your father's chromosome that he gave you in blue. Here's your mother's chromosome that she gave you in pink. Notice that they have the same types of genes. A, B, C, D, E, and F. And over here on the mother's A, B, C, D, E, and F. So that's how we represent the same type of gene by the same letter. But when there's a mutation that ultimately makes the nucleic acid sequence a little different, so you get brown hair here and blonde hair there, remember those are different alleles. So we illustrate this by capitalizing one allele and lowercasing the other allele to show you that the alleles are different. Well, notice in all of these cases, all of the alleles are different. You have capitals all on the fathers and lowercase on all the mothers. Okay, so that's part of the ease of explaining crossing over. So what ends up happening, and this is a random process, we don't understand exactly why it undergoes this at different places every time. It does not occur in the same place every time, and it doesn't have to occur, or it can occur multiple times between homologous chromosomes. So it's totally random as far as we understand it. And when we say something's random, it's usually because we don't understand what ultimately is doing the selecting. So they may cross over at this point one time. Next time they might cross over here and there and there. I mean, they can do it multiple times or they can do it not at all. It's just totally random. So what is crossing over? It's an equal exchange of genetic material between homologous chromosomes. Let me say that again. Equal exchange of genetic material between homologous chromosomes. So what ends up happening once crossing over is done is the chromosomes that result will all still have the same types of genes. Notice you don't lose A or B. You have A, B, C, D, E, and F on every chromosome. But now look what's happened. On this chromosome, you still have your father's original alleles that he gave you. Nothing's been recombined in that one. But on this one, you get 2 alleles from your mother's chromosome mixed with 4 alleles from your father's chromosome. That's a new combination of alleles that gets passed on. Over here are the 2 alleles of your father's that got traded with your mother's chromosome, and then here are the rest of your mother's alleles. That's a new recombination. And here, this is the original combination that your mother gave you when she donated her half of the genetics. Each one of these chromatids will end up in a different gamete by the end. And so, depending upon which one of these gametes is involved in the fertilization process, will ultimately determine what you pass on in that instance, in that recombination event, to your offspring. If your child gets this, they're going to have more of your father's features. If they get this, they're going to have more of your mother's features. If they get these, they're going to get some combination of both. And this happens across all 23 pairs. The only exception to this, again, is the male non-homologous sex chromosomes. Why do you think that crossing over doesn't occur between those? Because they're not homologous, and any crossing over would result in loss of genetic material. Now, with the females, the X chromosomes do cross over because they are homologous, and therefore you're not losing any genetic information. Now, sometimes mistakes happen, and you do get crossing over, but that ends up having some certain results where you could actually have an XX, but it develops into a male. We'll talk more about that in the next lecture. And it's not hermaphroditism. So we'll talk about ultimately some of the things that can go wrong and what happens. So you don't want crossing over to occur in here. It only happens if it's a mistake. But they do pair up because you do need to still separate them during meiosis at a later stage. Okay, that's the first recombination event, which is crossing over. So I'll ask you when that occurs, and I'll ask you to define crossing over. Again, here's just kind of the summary of it all. Homologous chromosomes exchange equal amounts of genetic information, and they create new allele combinations. Okay? Sometimes it can make mistakes and delete some of these things out, but again, those are mistakes. When everything's running fine, you don't lose any genetic information. You don't lose the genes for hair color or for eye color or anything like that. Okay? So what ends up happening, let's say the B was for hair color and the E was for eye color. Let's say your dad was blonde or brown hair, blue-eyed. Well, in this scenario, if you pass this gene on or this chromosome on, your child would inherit the brown hair, blue-eyed alleles. Let's say over here your mother was blonde hair. Now it's not this simple. Hair color and eye color are more complex than what I'm making them, but for the sake of an example, just simplifying it. Let's say your mother was blonde haired and blue-eyed. Well, here is the blonde hair allele, but you still have your father's brown eye allele. So if the child inherits this, they'll inherit the allele for blonde hair but brown eyes. Over here, you would inherit the child, if they inherited this chromosome, would have the brown hair allele and the blue-eyed allele. And over here, it would be blonde hair, blue-eyed. So that just kind of shows you how things can just get mixed and matched, and why when you pass those on, it's a random process of what gets passed on, and that's why siblings look differently, even from the same parents, because of this genetic recombination. Okay, now let's jump to metaphase 1. Another huge thing that happens here. This is the second form of genetic recombination. Yeah?
Real quick, do you believe that it's just random, or is it we just don't know?
They've shown that some parts are more likely to cross over than other parts. So there is something to why they pick certain locations, but we're not sure how that selection occurs. So it's like we know that twins are genetically, it runs in the family, but we're not quite sure exactly what does. So that's why when we say, it's a random process, it's usually because we don't know what ultimately is causing it. So it's going to remain a mystery for a while, but eventually we might figure out, 90% probability that it'll be in these parts right here because of this and this. And at that point, we start understanding it less as an ambiguous process, but say, hey, this is most likely what's going to happen, and make those predictions. So it's just about a lack of knowledge, ultimately. All right. Okay, let's see. Metaphase. So metaphase 1. Remember that in mitosis, all the chromosomes neatly line up along the middle. But a little something different happens in metaphase 1. They don't all line up individually. They line up still paired up with their homologous pair. So instead of lining up all 46 chromosomes along the middle, they line up as pairs along the middle. Now you may think, well, how is that genetic recombination? Because ultimately, what happens in anaphase 1, when the homologous chromosomes get separated from one another, how they line up here has everything to do with genetic recombination. We call this independent assortment. So, here's what independent assortment is. Here's the second mode of genetic recombination. And that occurs during metaphase 1. So here's an example. We're ignoring crossing over for the sake of this example as well. So here are the blue chromosomes, which are your father's half that he gave you. Here are your mother's chromosomes, which are the half that she gave you. As these homologous pairs line up along the middle, they don't have to line up the same way every time. So on this scenario, all the fathers are lined up on one side, all the mothers are lined up on another side. That's one possibility. Another possibility is these two chromosomes flip. This one goes to this side, this one goes to that side. Now, when these homologous chromosomes go into this side and these go into that side, you're going to get these alleles from your father's chromosomes and these alleles from your mother's chromosomes. So that's another form of genetic recombination. Now, this is not crossing over. This is just what chromosomes from what parents do you inherit? Do you inherit all of what your father gave you? I mean, do you pass on what your father gave you? Do you pass on what your mother gave you? That's one scenario. And notice there are all sorts of different ways in which these can flip to one side or another. Now, why is it called independent assortment? It's because when these two flip, it doesn't influence whether any of the other homologous pairs flip. So it doesn't matter which side they line up on, it's completely random, again, of which side the homologous pairs line up on. So, you know, in this scenario, this cell that results from it would have 2/3 of the father's genetics and 1/3 of the mother's, and so on and so forth. So that's independent assortment. Crossing over is able to assort alleles between homologous chromosomes. Independent assortment mixes up everything else. Now again, you don't lose any genetic information. Every person is going to, or every gamete that's produced is going to have all 23 chromosomes. It just depends upon what combination that your parents gave you do you pass on. Do you pass on 2/3 of your father's and 1/3 of your mother's? Half and half, most of your father's? That's why you start seeing children that say, oh, he looks more like your side of the family or more like your side of the family. So that's where independent assortment is genetic recombination. You're not mix and matching alleles between homologous chromosomes. You are literally scrambling up all of the homologous pairs that your parents gave you and passing on a different combination of those. All right. Now, anaphase 1. This is where homologous chromosomes separate. But notice the sister chromatids still remain attached. So during the first stage of meiosis, you don't have any separation of the sister chromatids. Remember, they're attached together by a centromere. So the sister chromatids remain attached during meiosis 1. All you do is separate the homologous pairs from one another. So that's anaphase 1. Homologous chromosomes separate. Now remember this one too, because later on when we talk about a process called nondisjunction, which is a problem that occurs later on in life, this is going to come up again. Now it's not a mode of recombination. It just separates the homologous pairs, but it is important for certain issues that we deal with later on in life. Now, telophase 1 and cytokinesis. Pretty much, again, we get into some differences here depending upon whether you're a male or female. Now, if you're male, if you produce sperm, cytokinesis will always occur. Okay? One cell will become 4 separate sperm. But with females, that's not the case. I'll talk a little bit more later on about how that works. But for now, we have telophase 1 and cytokinesis. So, same thing. Once you separate the homologous pairs, the mitotic spindle breaks down, nuclear envelope reforms. However, the chromosomes don't unwind at this point. Why do you think? They're going to go through another round of meiosis. So there's no point in unwinding just to ravel back up again. So, yeah, it doesn't happen during telophase 1. You don't get unraveling of the chromatin because there's still one more round to go. Now, you do get separation of the cytoplasm, which we call cytokinesis. This is for males. This doesn't happen in females. Okay? Now, meiosis 2 is pretty much the same as mitosis. So I'm just going to kind of breeze through this real quickly. You have almost like no questions on meiosis 2, but there is some part that you will have to know for one of the concepts, like I said. So let me just go through this. So prophase 2, remember the chromatin's already condensed. So the only two things that occur are the nuclear envelope breakdown and the mitotic spindle forms. Now notice you're starting with 2 cells here for male sperm production. Okay. Now metaphase 2, chromosomes line up, but notice these cells only have half of the genetics because the homologous pairs have been split up. So instead of starting out with 46 chromosomes here, you have 23 chromosomes in each one of these. So the 23 chromosomes line up along the middle. Now during anaphase 2, this is when the sister chromatids separate. But notice, too, that not even the sister chromatids are exactly identical to one another. Why? What has happened previously? Not independent assortment. Crossing over. because of crossing over, you've had some genetic recombination of the alleles, so not even sister chromatids are exactly the same anymore. That means that when these two cells separate here and these two separate here, each of these are going to have a unique assortment of alleles. No two gametes are ever going to be the same. Okay? So anaphase 2, sister chromatids separate from one another. And telophase 2, same thing. Cytokinesis, mitotic spindle breaks down, nuclear envelope reforms. And now you have 4 haploid, non-identical, meaning the allele combinations are unique to each of these gametes. I think on my quiz I say genetically dissimilar is the phrasing that I use. So they're genetically dissimilar. Why? Because of all of the crossing over and independent assortment that occurred back in meiosis 1. Okay. Now, I told you that there were 3 forms of genetic recombination. Anybody want to guess what the third is? So we have crossing over, we have independent assortment, the last one is fertilization itself. The sperm and the egg coming together. How is that genetic recombination? 2 different people's genetics coming and fusing into one. So that's the third. It happens obviously at the very, very end. All of these combined in the human genome can produce over 70 trillion unique combinations. So unlike what X-Files would tell you, you don't have a twin over in China or other planetary systems that may have humans on them as well. It's a very unlikely event. Now, if you've watched Jupiter Rising, then it can happen over enough. No, anyway. Just kind of give a spoiler, but oh well. It's a good movie anyway. Okay. So those are the 3 forms of genetic recombination. Crossing over, independent assortment, fertilization. Okay, so I'll ask you about these 3, as well as being able to describe them. Now, let me give you an overview of the difference between meiosis in males and female. Not the timing, but the end result. Because like I said, there are some unique differences that don't occur in females that do occur in males. The overall process of how the crossing over, independent assortment, and all of that happens is the same between males and females. The difference is what happens to the actual cell during meiosis 1 and 2. So for males, you get cytokinesis every time. So you get cytokinesis after meiosis 1, and you'll get cytokinesis after meiosis 2. In the end, for every 1 germline cell, which we call a spermatocyte, that undergoes meiosis, you're going to get 4 sperm. But in females, 1 oocyte, that's the germline cell for females, those are the eggs, 1 oocyte remains 1 oocyte. You're like, well, how does that happen when you have all of the same things that happen in the males? Well, here's the thing. When these nuclei form during meiosis 1, instead of undergoing cytokinesis, the egg simply throws out one of the nuclei. So one of them will remain with the egg, the other one gets ejected out of the egg. So no cytokinesis occurs. Now, whichever one gets left in there will go through the second stage of meiosis. And again, it'll choose one of the 2, and the other one will be thrown out at the very end. So in the end, 1 egg will always remain 1 egg during meiosis. You get no cytokinesis, but you get the same process of what's happening with the genetic recombination. So women will produce 4 nuclei, but there will only be 1 egg. Now the reason for this is because there are proteins and other things in the cytoplasm that you just can't split up. Okay, so the egg needs to remain intact with all of what we call maternal components. And a lot of those are for getting the metabolism started and fusing the nuclei and other things like that. So that egg, all of what starts in this egg, needs to remain in there. The only thing the cell doesn't need is the extra nuclei, and it just retains 1. Here was the other thing I was telling you about. When a woman ovulates, the egg, the oocyte, is actually right about metaphase 2. And only when the sperm fertilizes the egg will it finish meiosis II and get rid of the second nuclei from meiosis II. So you essentially have the sperm coming in one end. Once it comes in, this will finish off and throw out the egg, or throw out the nucleus from the other. And then the two nuclei fuse and you have the zygote, or that stem cell that becomes you. So that's the thing about meiosis. As I mentioned, meiosis begins before a woman's even born, continues on through their life, and is about meiosis II when the egg ovulates, and or halfway through meiosis II, and won't finish it unless fertilization occurs. If it doesn't occur, then it just stays at meiosis II, the egg goes through, and you don't get pregnant. Okay, now, twins. Monozygotic versus dizygotic. All right, so monozygotic or identical twins. The reason why they're identical is because they come from a single fertilization event. If you remember, I told you about how after several cell divisions, you end up having what we call a blastocyst, where it has all of these cells on the outside that will become the placenta and the amniotic sac, and they have these cells inside called the inner cell mass. That's what becomes you, that group of cells right there. Well, these cells in the earliest stages have the potential to become every cell in the human body. And we're still trying to figure out all the reasons why, but on occasion, the cell mass splits in two. Now, because all of those cells came about through the process of mitosis, the genetics on all the cells are exactly the same, which is why identical twins have the same genetics. Okay, so that's why we call it monozygotic. It's because it's from a single fertilization event, but then it splits into two inner cell masses, and you end up having identical twins. Now, dizygotic twins are two separate fertilization events. When a woman ovulates two eggs instead of one, and that happens quite readily when you have hormones or other drugs that will stimulate that for someone who maybe is going through a hard time getting pregnant or whatnot. Ultimately, if you release two eggs, there's more than enough sperm in every ejaculation. There's like 10 million sperm or whatnot to fertilize both of the eggs. So it's not a question of how much sperm, it's a question of how many eggs the woman ovulates. So if two eggs ovulate, each one of those have gone through their own meiosis. And the sperm obviously have gone through their own meiosis as well, which means that they're going to be no different than any two siblings, brother and brother, sister and sister, or brother and sister. This is why fraternal twins can be different sexes, because you either fertilize it with an X chromosome or with the Y chromosome. Here, however, identical twins must be the same sex, because it's a single fertilization event. You're not getting two different sets of sex chromosomes. Now, I'm sure there's an exception somewhere in the world of a 1 in a trillion chance that this happens and whatnot, but I won't go into how that occurs. So, now, problems do arise, especially as you get older. Now, they occur more often in women because, if you remember, I told you that women will make all of the oocytes they'll have for their entire life before they're even born. Whereas men won't even start to use their gametes, their germline cells, to create the gametes until they reach sexual maturity. So there's this offset of at least a decade, maybe a little bit more, in which women start, using the eggs versus men producing them. As such, we usually track this problem in women because it's more likely that the man is about the same age as the woman. You're not having a guy in his 70s having kids with someone in her 20s, although it happens. So the reason why I say this is because this occurs in men as well, but it doesn't occur until we're in our 60s or 70s with as much frequency as what it occurs in women's in their 40s. Okay? It's called nondisjunction. Now, before we get to nondisjunction, let me explain one major issue that that sometimes happens but doesn't come to term. In fact, this is how almost 20% of miscarriages occur because of this problem, and it's called polyploidy. Polyploidy is when more than one sperm typically fertilizes the egg. Now, there are usually mechanisms in place in the egg to prevent what we call polyspermy, because what ends up happening is you end up getting 3 of every chromosome type instead of 2. And the cell needs to have exactly 46 to really work. Any more or any less chromosomes than this, and there usually are major problems, if not fatal problems, in the development of the embryo. There are always exceptions, but the general rule is any deviation from the 23 sets of chromosomes that we have in our cells results in huge problems, resulting in miscarriage. So, an extra set of all of your chromosomes is a big problem. Now, sometimes plants can actually overcome this issue, but for animals, it just doesn't happen. Okay, so that's polyploidy. However, what we're going to focus on is what's called nondisjunction, mainly because this is where the exceptions to the rule come into play. So what is nondisjunction? Well, it can occur in one of 2 phases. It can occur in either anaphase 1 or anaphase 2. And this is where I told you that these 2 processes will come into play. So what is nondisjunction? Well, remember in anaphase 1 or anaphase 2, you get the separation of homologous chromosomes or the separation of sister chromatids, okay, respectively, anaphase 1 and anaphase 2. As the cells get older, it becomes more likely that when this process of anaphase 1 occurs, or anaphase 2, that the homologous pairs don't separate. And so when that happens, or in anaphase 2 it would be the sister chromatids don't separate, and I'll show you an example right after that. When that happens, the resulting gametes at the end up having an extra or 1 less chromosome. So instead of having 23 chromosomes, they end up having 24 chromosomes. Or they have 22 chromosomes, 1 more or 1 less. So here's an example of what would happen if it occurred in anaphase 1. Let's say, now it's ignoring all of the other chromosome pairs. So it doesn't mean there's nothing in this cell. It just means with this chromosome pair, none of them went over to this cell. Okay? But all of the others separated just fine. So that's what this is illustrating. So what ends up happening is when these chromosomes go into one cell and they don't split as they should, that's called nondisjunction. Then when these split during anaphase 2, half of the gametes, like let's say these are the oocytes, because those are usually what we track is in women, half of the gametes will have 1 extra chromosome. And over here, half of the gametes will have 1 less chromosome. Now, in almost all cases, this is fatal. The embryo cannot survive. Let's look at another example. Here, in anaphase 1, you get separation just fine. Okay, no problem. However, in this one right here, the sister chromatids don't separate properly during anaphase 2. And so what you get is this one has 1 less, this one has 1 more. These 2, they're fine. So you only have half of the gametes affected by this if it occurs in anaphase 2. Whenever it occurs, anaphase 1 or anaphase 2, if you have 1 more or 1 less, like I said, it's almost always fatal. Now there are exceptions. For example, chromosome number 21. It's one of the smallest chromosomes that there is. And that's the reason why this has the highest survivability rate, about 85% survivability. And this is what results in Down syndrome. Down syndrome is when the child has a normal set of all chromosome pairs, but they end up having an extra chromosome 21. Okay, so what ends up happening is this is why women over 40, they actually have 10 times the chance of having a child with Down syndrome than women under 40. Now, it's not that you reach 40 and all of a sudden it's 10 times. They just split up the groups, and when women are in their last few years of fertilization, they are 10 times more likely to have this problem occur in their production of the gametes. Now, this can occur in males as well, like I said, but usually not until a couple decades later. So it happens in males as well, but we mostly look at females. Now, there are others that are less common, but they only have like a 5% survival rate. And these are, trisomy 18 and 13. So if we go back to the karyotype, that would be an extra 13 and an extra 18. But those have a very low survivability rate. And all of the others in the autosomes that we see, pretty much the fetus aborts. Now, that brings us to another concept, trisomy. What does trisomy mean? Trisomy means you have 3 of a homologous group rather than 2. Okay? Monosomy means you're missing one. So in the case of here, this is trisomy, but when one cell loses a chromatid, then you end up having what we call monosomy, where you only have one of the chromosome pairs instead of both of them. Okay, now, this is for the autosomes. What's interesting is it is actually fairly likely for the embryo to survive if it occurs in the sex chromosomes. So the sex chromosomes are less stringent about having an extra chromosome. In fact, in some cases, it makes no difference whatsoever. For example, Jacob's syndrome. About 17% of males have an extra Y chromosome, and it doesn't do anything. It doesn't make you more aggressive, as they once thought, because really they only saw this phenomena when they were taking the DNA of men in jail, and they noticed a very high percentage. Well, then they started sampling the general populace as a whole of men that weren't in jail and found the same statistics. So it led them to believe at first that males with an extra Y chromosome tended to be more aggressive, committed crimes, and other stuff like that, and that's not true. So men can have an extra Y chromosome and be just fine. Why? I mean, look how small that thing is. It's just got a few hundred genes on it, and most of the time an extra one of those really won't cause any problems. Now, Klinefelter syndrome, XXY, this is still a male, and in a lot of cases, again, they're fully normal. Now, this is not a hermaphrodite, okay? It doesn't even lead to that. There are situations where men can have underdeveloped testes or slight breast development, but that's not hermaphroditism. So I'll explain hermaphroditism later. But XXY, in fact, a good portion of men can have an extra X chromosome and be just fine as well. So it all depends. There's some dynamics during the development process that really make or break whether or not you're going to have certain problems or not. Now, Triplo X syndrome is a female because there's no Y chromosome. So pretty much you get a Y chromosome, you're going to develop male. That's just how it is. But if there's no Y chromosome, you develop female. So 3 X chromosomes, there are certain abnormalities with this. There are certain genetic defects from infertility to some other aspects. But it's actually fairly common too. To have 3 X chromosomes, and the survivability is really high. Now, the only case of monosomy that we've ever seen, remember monosomy means one of the homologous pairs, is what we call Turner syndrome. This is where you have a female with just 1 X chromosome. Now, it has a 1% survivability rate, and they're always infertile, so it's not very common. This is the only case, though, of monosomy that we've ever seen where the female survives to be born and mature. Okay, but it's always infertile. There's always some type of mental retardation and developmental retardation, especially in the sex characteristics and whatnot. So that's just that. Now, with men, you've got to have an X chromosome. Remember, there are over 1,000 genes on this. That's 5% of your genome. You don't have an X chromosome, you don't develop. So there's never been a case of monosomy male with no X chromosome. Okay? So these are just some of the examples of trisomy, where you have 3 of a chromosome pair, in this case the sex chromosomes. And this is the only case of monosomy. So really, Down syndrome is the most prevalent case of what we call autosomal nondisjunction. These are in the non-sex chromosomes. Okay? So that's one of those that I'll test you on as far as what trisomy 21 causes and whatnot, and what trisomy is. Okay? But you'll also have another question on defining nondisjunction.